This, and we will be adding this onto our social media platforms. So you're welcome to have your video on or off based off your privacy, okay? Cool. All right, so hello and welcome everyone to MSA SPS is first Student Mycology Symposium. We are awesome. All righty, so our next speaker is Robert Blundell. So he is a plant pathology master's student at the University of California, Davis, and he's studying the sustainable methods for grapevine trunk disease control with Dr. Akif Eskelin. So Robert grew up in the UK, where he received his bachelor's degree in biological sciences at the University of East Anglia before going on to work with the John Innes Centre and the British Sugar Beet Research Organization. In 2016, he moved to the US to work with the North Dakota State University's Sugar Beet Pathology Lab with Dr. Melvin Bolton. Switching to Davis in 2017, he moved to Dr. Claire Castile's lab at UC Davis to study the effect of organic management on tomato health and resistance to insects, where he's published in Nature Plants. Robert is now in the final year of his master's program working under Dr. Akif Eskelin, enjoying working with beneficial fungi and pathogenic fungi to mitigate grapevine trunk disease. And when he's not in the lab, you can find him backpacking in the Sierra or mountain bike racing. All right, go ahead, Robert.
Perfect. Thank you, Tanya, for that introduction. And thank you, Maria and Gillian, and everybody else involved in setting up this event, as well as coordinating it. I really appreciate the opportunity to present to you guys today. This is the first time I'm presenting my master's research, so it's an exciting day. So my title is Discovery, Identification, and Implementation of Potential Biological Control Agents for Pruning Room Protection against Grapevine Trunk Diseases. So first, I thought we'd start with a little history, as archaeological evidence suggests that humans began using and enjoying the products of grapes way back to the Neolithic era, around 6,500 to 7,000 BC. And a little after that, grapes actually began to be cultivated in what is now modern-day Georgia. So nowadays, grapevine production has a huge global reach, with more and more new countries, such as Scandinavia, recently adopting cultivation of grapes. And ultimately, this has led to grapes being the highest production fruit in the world per ton. The main products we get from grapes are wine, table grapes, and raisins. And here in California, grapevine production is a pillar as well as an icon of its economy and agricultural identity, with about 90% of US grapes being grown here in California. And the growth acreage is largely split up into these key growing regions. So we have the North Coast, which includes world famous Napa Valley. We have the Central Coast, San Luis Obispo and Santa Barbara. We also have the Southern California desert growing region. And we have the Central Valley, which is where the majority of table grape production is. However, grapevine production in California is constantly under threat. For example, here you can see the glassy winged sharpshooter, which is a hemipteran that vectors the virus responsible for Pierce's disease, which is catastrophic for grapes and fortunately is not too much of an issue yet here in California. Hopefully it stays that way. And here you can see the fungal disease powdery mildew, which plagues growing regions worldwide. And also, not to mention another threat to the wine industry, which is the craft beer drinker. So the focus of my talk to you guys today is one of the most serious threats to viticulture production worldwide, and this is the grapevine trunk diseases. And actually, for the last 3 decades, they have been the most damaging grapevine disease. And the most accurate way to refer to grapevine trunk diseases is as a complex, with over 60 fungal species responsible, with multiple species responsible for each disease of the complex. And the main diseases of the grapevine trunk disease complex, we have ESCA, which actually in its own right is its own complex, and we have Botrisphaeria dieback, and we also have Eutypa dieback. So there are also minor members of the complex, such as Phomopsis dieback, but generally I won't be discussing these too much today. Now, these diseases are caused by taxonomically unrelated fungi, mainly belonging to the Ascomycetes, as well as some Basidiomycetes. And the main way that these fungi gain entry into grapevines is via pruning wounds, which is an important yearly practice, which generally happens anytime after harvest, through winter into early spring. And after entering, they colonize the xylem, or they ultimately become wood pathogens. permanent wood pathogens. And they're considered slow progression diseases because they often take several years to manifest and show their symptoms after that initial infection event. And often the symptoms are inconsistent. So this therefore is obviously quite a challenge to control, because often when the symptoms show, the vine has already been compromised and grapevine death has already begun. So shown here are some of the main symptoms that we associate with grapevine trunk diseases. You can see both Botrytis dieback and Eutypa dieback, as their name suggests, with dieback have very obvious symptoms of dieback, so a loss of vegetative shoots, particularly on whole arms. And because grapevine trunk diseases are wood pathogens, if you look inside the wood, it's easier for us to diagnose in the field, you're going to see cankers. And for Eutypa dieback in particular, they're often characteristically wedge-shaped. So leaf chlorosis is another common sign that these guys are present. So eutypine is a phytotoxin that is produced by Eutypa larda, which is the main causal agent of Eutypa dieback. And this toxin in particular is responsible for the chlorosis associated with this disease. And furthermore, with berries, we see wilting and ultimately a big yield reduction. So these diseases pretty much result in complete death eventually of the vine by irreversible loss of function of the xylem. And the economic fallout for growers is pretty much an increased disease management cost. And sadly, some growers will not actually get a return on their investment if their vineyard suffers a heavy infection. And from the graph here, you can see that vineyard longevity decreases severely as inoculum and infection events occur.
Hey, Robert, sorry, real quick. Do you mind coming a little closer to your screen?
Oh,
because
sure.
there seems to be some sound that's fading and we can't pick up all your words.
Oh, okay, not a problem. Try.
Oh, I don't know if that's the case then, because now you got closer and it's still kind of weird. Okay,
Okay.
we'll push through.
Just try without headphones, maybe. Doesn't look like that. We'll go back to this. Hope this is okay. So as well as being a major problem in California, they affect pretty much every other grapevine growing region worldwide. And of the grapevine trunk diseases, Botrytis and Eutypa dieback are particularly damaging for growers in California, and our research concerns their control. So 23 Botrytis species have been identified as causal agents, and the most aggressive of these is Neophysococcum parvum, with pygmidiospores being the main source of inoculum. And for Eutypa dieback, we have multiple causal species in the Diatropiaceae family, with Eutypa larda, as I mentioned, being the most virulent of these. And for Eutypa larda, we have ascospores released from an ascus, which overwinter in Paraticia. And we should also mention that these grapevine trunk diseases, whilst being studied for a very long time, it's always a new challenge. For example, that was the first report of this new fungal agent in Chile just a couple of days ago, actually. So in terms of sources of grapevine trunk disease inoculum, there are quite a few. A lot of material for grapevines comes from nurseries, and if it's infected, that's a pretty easy way of just moving that inoculum around to new fields. There are different hosts as well, which can host these grapevine trunk diseases. So if you have one of those growing in a nearby field, that can be an issue. Also, mummified berries left over from the previous season often get left on the vineyard floors, and that's a pretty big source. They may simply just reside endophytically, but in a non-parasitic way, and at some point they might, due to acute, turn parasitic. And generally, pruning and harvesting debris, a lot of vineyards don't invest the time to kind of clean it up because there's a lot of work, so that's often a source of inoculum too. So these grapevine trunk diseases use wounds, including natural and human-made, to enter the grapevine vascular system, colonizing the host and causing wood decay. As I mentioned, pruning wounds are the most common route of entry. So pruning involves trimming back the canes to prepare for the new season's growth, and it's pretty much performed anytime after harvest through spring. And unfortunately, pruning timing generally overlaps with precipitation events, which mobilizes fungal spores. So rain and wind disperse the spores and they land on fresh pruning wounds. When conditions are conducive, the spores can colonize the wound and enter the xylem. So in terms of management, controlling grapevine trunk diseases is very challenging because I mentioned they often spread in the vine before showing symptoms. And once infection occurs, there are no good curative strategies available. So as a result, preventative practices are pretty much our only good approach, and they can be classed as either cultural or as wound protectants. So one good cultural practice is safer pruning, whereby you sanitize pruning shears regularly and you prune, hopefully in a dry weather window, when the risk of spore release is at its lowest. Another approach in California is delayed pruning, and this can be normally February or later, or as close to bud break as possible, because this takes advantage of reduced wound susceptibility to infection, as the majority of rainfall events have already occurred, and the weather is warming up, which studies suggest that this helps the wounds heal quicker. Double pruning is also an option for some vineyards. When crew availability is high, they can do an early pruning, and then later on, just before bud break, a second pruning can be performed to remove any infected material, but that is quite an expensive process. So the next main way to limit grapevine trunk diseases is via application of a pruning wound protectant, and these can be pastes, but are more commonly liquid sprays. So chemical pruning wound protectants have been extensively studied and used, but some effective chemicals have been banned over environmental concerns and human health concerns, such as sodium arsenite, for example, which was pretty effective for a long time as the only good control of Esker disease, but this has now been banned. And generally, chemicals get a bad rep for there's studies suggesting destruction of natural microbiota as well as harming soil health. So we desperately need to develop sustainable wound protectants. And the main focus of sustainable wound protectants is the use of biological control agents involving using naturally occurring microbes, most commonly bacteria, and fungi with antifungal properties. And the majority of studies to date have shown that Trichoderma species, as well as Bacillus species of bacteria, are the most effective. Apart from the environmental benefit I just mentioned, the other main reason why we're interested in using biological control instead of chemicals is because once you create your pruning wound, it's quite a long period of susceptibility where those grapevine trunk diseases can enter. And if you spray a chemical, you're likely just going to get short-term protection. But we hope that some of these biological control agents should be able to colonize and stay on that wound and provide long-term protection. So Trichoderma species account for an active ingredient in more than 60% of today's registered biopesticides, and this is largely due to their ubiquitous presence in nature, as well as multiple modes of biological control, including antibiosis, competition for space, as well as inducing a plant defense response. And for bacteria, Bacillus subtilis has been the most successful, largely due to antibiosis. So now getting on to our latest research in our lab regarding this. Whilst there have been plenty of in vitro lab studies testing biological protectants, there are comparatively few field level studies that take them through to this stage. And this is the data that the growers really need to assess and base their decisions off whether they're going to want to use biological control over chemical if they're going to use a protectant at all. Also, the majority of field studies have focused on controlling U-type dieback and Esker, but Botrytis dieback has comparatively few studies. So our approach to furthering knowledge regarding this was firstly to investigate whether grapevine sap could be a reservoir for antagonistic microbes, and if we could isolate them, we would evaluate them in vitro in greenhouse and in field trials, and we would compare them to existing registered biologicals and chemical options. So we're particularly interested in looking for BCAs in grapevine sap because this hasn't been described in the literature yet. And sap is a very nutrient-rich source, so we predicted it might be quite a conducive environment for microbes to thrive, especially as it's the first line of defense for the plant as well. So you could predict there might be some beneficials in there. We also wanted to try and source some potential BCAs from elsewhere as well. So in the fall, we split open some canes after harvest that looked healthy, and we made some isolations from the tip of the cane as well. So after making clean subcultures of isolates, we extracted DNA and performed sequencing to the genus level to identify them. This is just kind of a pick of what we actually isolated. There were plenty more, but some were pathogens as well. So largely we're culturing Orobasidium, which has been mentioned a lot in the literature, but we did also isolate Trichoderma and Bacillus. And there are pretty much two main in vitro antagonism assays we did firstly is dual culture, which is a simple test where you would put a mycelial plug of your isolate and then of your pathogen on the same plate, and the control plate consists of just the pathogen, and after a set number of days, you record the radius and then compare the radius in the antagonism, and that will kind of tell you how much inhibition it's creating. And we pretty much found that the trichoderma and the bacillus were able to cause inhibition of E-type Alata in vitro, and just trichoderma was able to cause inhibition of Neophysical compartment. And when we look at this visual representation of the graphs, we see some different mechanisms in our assays. So we can see that against E-type Alata, trichoderma, for example, is causing overgrowth as its mechanism of inhibition, just completely overgrowing it. Whereas for the faster growing pathogens such as parvum, we're seeing stopped growth where they kind of meet in the middle. And for bacillus, we actually saw what looks like potentially some antibiosis as well. And then pretty much the isolates highlighted in green, we took forward to Orobasidiums, to bacillus, and to trichodermas for subsequent studies. So we also included another Botryosphaeria diabag pathogen and Ampelina as well for Mopsis. So the second antagonism assay is a volatile organic compound assay to kind of see if any of these isolates are giving off a volatile which is causing inhibition that we wouldn't have seen in the previous assay. So in this assay, you just use an isolate plug similar to in the previous one on the center of a base plate, and then you put the pathogen on another base plate, sealing them together. And interestingly, this time, so for the trichoderma, which is 87178368, in the dual culture assay, it was overgrowing it, but here we're also seeing inhibition when they're not obviously touching. So this suggests that trichodermas may also be secreting or giving off a volatile. And unfortunately, we weren't able to get to the point of extracting and analyzing these volatiles, but that would be useful in the future. And then for those Orobasidium, bacillus, and trichodermas, we perform multilocus identification to the species level. And we found the Orobasidium were Paludolans. We had Bacillus thalazensis identified as the closest species, and for trichoderma, we had Trichoderma asperellum and Trichoderma hamartum. So now moving on, we took these isolates to test them as pruning room protectants in a greenhouse detached cane assay. So pretty much this is where we took canes from the field and we cut them to be about 10 centimeters long, single node length. And on the day of the experiment, we made a cut 1 centimeter below the tip. And this would represent a field pruning room, for example. And then we would spray the protectant onto these canes. And here's a quick overview of all of the different treatments we used. Here's our experimental ones here versus registered chemical and biological treatments. So after spraying them, we came back a day later and we inoculated the canes with a spore suspension of either Neophysical compartment or E-type Alata. And then after 4 weeks, we came back and we split these canes down the center and we performed, we took isolation from segments from this region to grow it on a Petri dish to measure the recovery of the pathogen, as well as if we could actually recover that original biological control agent. So just to give a quick overview of the results, we recorded it as mean percentage disease control. And generally, we saw that the majority of treatments could perform better against E-type Alata than parvum, likely due to the fact that Neophysical parvum is a faster and more aggressive isolate, faster growing. And this represents when we performed multiple inoculations. So for a subset of canes, we just did one inoculation event, and then for another subset, they had two. So one at 24 hours, one at 1 week, and the other one had 24 hours, 1 week, and 2 weeks, thus representing rain events in the field, which would spread inoculum. And we generally saw that as we applied more inoculum, the efficacy of them did appear to decrease, but some of them still performing quite well. And trichoderma products in particular, I should say, performing very well. For biological recovery, we noticed that the trichoderma isolates have very good recovery, and often, even when we performed multiple inoculation events, the recovery didn't decrease too much, though obviously the recovery doesn't appear to correlate in all aspects with disease control. For example, bacillus of BCA5 and GCM, for example, they had some low recovery, but actually some high disease control. So there is a lot of variation. And then moving on to our 2020 field trials, which is kind of the ultimate test to see how well these protectants do. So using exactly the same treatment list, we performed a trial in Sacramento County and Kern County. And this, we pruned vines in February of 2020, and then we would apply the treatments, and then the next day we'd be applying the pathogen. Just a quick overview. I should say that once we sprayed the protectants, we applied the chemical treatments 1 day later with the pathogen, but we came back 7 days later for the biologicals to give them time to colonize to properly assess how well they could perform in the good situation. And then we came back in October 2020 to collect those treated canes. So we left them for 8 months in the field, wait until after harvest. And like we did before, we would cut down the center of the cane to make our isolations to monitor whether we could recover that pathogen and that biological control agent. So firstly, for Sacramento County, our trial revealed that the biological protectant biotin, which is shown here, which is now approved and a registered chemical, was very effective again at protecting pruning wounds, both against Inoculum and as well against Neophytococcus parvum here. This is particularly important and promising results because simultaneous disease control with a single treatment means reduced labor and costs from the grower. Of the two chemicals included in this study, Toxin N, and Rally, which is kind of the industry standard, also performed well, but we're also seeing quite a few biological treatments also performing as well, if not better. So this study also highlights that the efficacy of protectants varies depending on the pathogen. For example, we can see that lunar sensation performed well against Inoculum and parvum. Our isolates did perform encouragingly, but likely not as well as the commercial ones, due to the commercial ones have been optimized for protectant with having adjuvants. And then in Kern County, we generally saw variation, but also some similarity to our trial in Kern County. Biotin again was the best performing, protecting against Inoculum. It didn't perform as well against parvum. It was beaten by Serenade and Vintec. Vintec also being another trichoderma, but largely biologicals also performing very well. We did see for the inoculated control, we did see only about 25% recovery, which is quite low, unfortunately. So in subsequent years, we'll hope to optimize that and get a better inoculated control to better assess how well these perform. But definitely between the two trial sites, there is variation that can explain these results, such as because we set these up 1 week apart and it was a little bit drier in Kern County the following week, which is not as conducive for the pathogens to develop. Quickly looking at recovery, again, we got some really encouraging results with this, that trichoderma products, Vintec, being a trichoderma based, had a very good recovery. Again, we're seeing that bacterial protectants had a poor recovery, as well as Oribasidium, though not a very good protectant, did have quite a good recovery. And then finally, just going to end by talking about another study that we're currently investigating, and this is looking at the effect of pruning timing in the dormant season on colonization of pruning wounds naturally by trunk diseases and beneficial microbes, and whether there's any particular timing of pruning that will be more conducive for the bacterial, for the beneficial microbes to be present versus the grapevine trunk diseases. So as a conclusion and outlook, trunk diseases are a huge threat to viticulture production worldwide, with pruning wounds being the major point of entry for these diseases. Biological control agents can be a sustainable preventative control technique, and they can perhaps perform as well, if not better than chemicals in some situations. And it does look encouragingly like they could colonize the pruning wounds for the duration of a growing season. Ultimately, fewer sprays will mean less money spent for growers, but we will need to confirm the efficacy over multiple years of field data. As encouraging as our data was, one year is just not good enough. And growers largely base decisions from field trial data. Thank you very much for listening. I'd like to thank Dr. Akiva Scarlin, my PI and mentor, for this, for taking me into his lab. Thank you to Molly as well, and Tim and Paul, who are all undergraduates who were a big help setting up these field trials and helping to analyze the thousands of canes that we had to go through. And happy to take any questions. Oh, and thank you to Sonoma County Mycological Association, as well as the American Society of Ethnology and Viticulture for providing some funding, as well as these 2 awards from UC Davis. Thank you.
Awesome. Thank you so much, Robert. One quick thing, maybe don't use these headphones next time.
Sure, that's good to know. Yeah, I'm sorry about that. That's
yeah.
Now.
Yeah, no worries. Hopefully people were able to follow along well. But we have a couple of questions. So I'm going to go ahead and read Rebecca's question for you. Were any of your isolated fungi also pathogens? A lab mate did a study where she found a lot of the best biological control fungal isolates were minor pathogens.
Yeah, that's a good question, a good point. We also did find that. We found a lot of Fusarium we were recovering to be fairly, as well as Epicoccum to be quite effective in some of our assays, but some pathogenicity trials that I didn't show, they do appear to be more pathogenic than useful at this point, so we didn't include those. But the vast majority did seem to be beneficial at least, but as we are taking them from natural canes in the field, we definitely did isolate some pathogens.
Okay, thank you. Casey, I see you have a question. Do you want to unmute and say it, or would you like me to read it for you?
That's fine, I can read it.
Awesome.
Okay, so yeah, I was just wondering, are there particular cultivars or rootstocks that you are working with in your studies, or because trunk disease is so pervasive, does it really not matter?
Yeah, it's a good question. You're pretty much right, yeah, because they are so widespread and pervasive. There's no real particular cultivars, especially in California, that have much resistance. I think in Europe, where they use Malo quite a bit, I think that's quite effective, or sorry, not very susceptible to ESCA. But against U-typer and Botrytis dieback, there's not really any particular rootstocks that have been shown to have significant control against them, unfortunately.
Thank you. Now, Brian, I see you have a question. Do you want to go ahead and unmute and speak, or I can read it for you?
Sure.
Okay.
Great talk. Thanks so much. I had just a minor technical question on one of your earlier slides. When you did the volatile inhibition assay and you had the inverted plates, was there anything in between them?
There was a sheet of parafilm.
Okay, cool. I thought that I missed that, but I was just curious about that.
Yes, sorry.
Really elegant assay. I love it.
Thank you. Yes, forgot to kind of include that in the visual.
Any other questions? Okay, that's not the case. Then thank you so much, Robert and Daisy. We really appreciate you two being the first speakers for our virtual symposium. I forgot to introduce myself. I am Tanya. I'm the vice chair. We have Maria here, who is the communication, or not communication, excuse me, you're the webmaster. We have Jillian, who is the communication chair. We have Rebecca, who's the past chair. We have Casey, who is, I forget, were you the secretary? Yes, okay, sorry, you're the secretary. And then I think Rob is here somewhere too, but he is the chair. So we want to thank you all. And if possible, we want to take sort of a screenshot of all of us. If you can turn on your video, cool. If not, don't worry about it. But we do want to include this in our social media as like our first foray into virtual symposia. Well, all right, we got a couple of folks here. So there might be two. Okay, all right, well, now it's just one page because people left. Cool. Awesome. Well, thank you so much. I'm going to go ahead and screenshot this. And everyone smile. Okay, 1, 2. All righty. Awesome. Thanks so much. And have a good weekend, everybody. I'll see you next month.
Thank you.
Thank you.
Everyone.
Thank you.